Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Ria Kakad, Editorial Assistant at Drug Target Review. Today's speakers will be Dr. Yong Zhao, Principal Scientist and Study Director at Eurofins Discovery. Following his presentation, we will move on to a live question and answer session, where you can ask any question to today's speaker. Please remember, you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to Yong.
Thank you for the introduction. Thank you for attending my presentation today. My name is Yong Zhao. I'm a principal scientist and study director for ADME-Toxicology services at Eurofins Discovery. Today, I'm going to talk about the transporter-mediated drug-drug interaction, in short, DDI. I will be talking in details about what in vitro assays are available to evaluate transporter-mediated DDI in drug discovery program and how Eurofins Discovery can help you assess transporter-mediated DDI. And this is the outline of my presentation today. First, I will give you a brief introduction of transporters and how transporters are involved in DDI, why evaluating transporter-mediated DDI is important in drug discovery. Then I will describe what regulatory agencies' recommendations are on transporter-mediated DDI. What are the available in vitro test systems for evaluating transporter-mediated DDI? At Eurofins Discovery, we have developed and validated a comprehensive panel of in vitro assays to evaluate transporter-mediated DDI. I will talk in details about those assays and the utility of the assays in assessing transporter-mediated DDI to meet regulatory agencies' recommendations. I hope after my presentation, I will provide you with useful information on what in vitro assays are important and how to choose those assays to assess transporter-mediated DDI. At the end, I will give you a brief overview of our company, Eurofins Scientific, Eurofins Discovery, and ADME-Toxicology services at Eurofins, and leave about 10 to 15 minutes to answer any questions that you may have and have further discussion on this topic. What are the drug-drug interactions and how are transporters involved? DDI can be defined as the change in a drug's effect on the body when the drug is taken together with a second drug. A drug-drug interaction can delay, decrease, or enhance absorption of either drug. This can decrease or increase the action of either or both drugs or cause adverse effect. DDI is mediated by two major pathways, transporters and or drug metabolizing enzymes such as cytochrome P450. Today, I will focus on transporter-mediated DDI. Transporters are transmembrane proteins that facilitate a cross-membrane movement of ions or molecules, including endogenous or exogenous substances, such as toxins and drugs. Here is an illustration of the SLC transporters on the cell membrane. Transporters are expressed in different organs throughout the human body, including intestine, liver, kidney, and the blood-brain barrier. They control the access of membrane-impermeable substance to different sites in the body. Research found that many pharmaceuticals interact with drug transporters. The drugs either need the transporter's help to get in or out of the cells. In this case, those drugs are substrate of the transporters, or they inhibit transporters' activity of moving other substrates. In this case, they are the inhibitors of the transporters. This interaction may lead to clinical relevant drug interactions when patients take multiple medications at the same time. Because of those interactions, drug transporters contribute to variability of the drug concentration and response, may cause unexpected toxicity or DDI. Here is one example of transporter-mediated DDI and how the drugs involved are labeled based on the knowledge of the mode of action in the DDI. The DDI here is between cyclosporine and Lipitor. Cyclosporine belongs to a class of drugs known as immunosuppressants. It is used to prevent organ rejection in people who have received the liver, kidney, or heart transplant. It is usually taken along with other medications to allow the new organ to function normally. Lipitor belongs to a group of medicines called HMG-CoA reductase inhibitors, or statins. It reduces the amount of cholesterol in the blood. It is discovered that cyclosporine can increase the exposure of Lipitor. Further research found that cyclosporine is an inhibitor of OATP1B1 and OATP1B3, which are liver drug uptake transporters. Lipitor is a substrate of OATP1B1, through which it is absorbed into the hepatocytes for clearance. As illustrated here, the blue dots represent Lipitor, which are absorbed into the hepatocytes through OATP1B1 for clearance. When transporter OATP1B1 is inhibited by cyclosporine, the uptake of the Lipitor into the hepatocytes is reduced and leads to the increase of the bioavailability of Lipitor in the blood. The normal dosage range of Lipitor is 10 to 80 milligrams per day. Due to this DDI, there is a warning on the drug label of Lipitor. The warning states, in case where a co-administration of Lipitor with cyclosporine is necessary, the dose of Lipitor should not exceed 10 milligrams. Let's talk about transporters that are involved in DDI. The drug transporters are generally divided into two families: ATP binding cassette transporters, in short ABC transporters, and solute carrier transporters, in short SLC transporters. The ABC transporters uses ATP as driving energy to export the drug out of cells. Therefore, they are also called efflux transporters. There are about 50 subtypes of ABC transporters in 7 subfamilies, including some very important transporters involving in DDI, such as multi-drug resistance 1, MDR1, or P-glycoprotein, PGP, breast cancer resistance protein, BCRP, and multi-drug resistance associated protein, MRP. ABC transporters expressed in small intestine, liver, and kidney. SLC transporter transport their substrate from outside to inside of the cells. They are also called uptake or influx transporters. Some of the SLC transporters, such as MACE, are bidirectional. SLC transporters move their substrates either by passive diffusion along its concentration gradient, or by co-transport or counter-transport against its concentration gradient by using the concentration gradient of another solute. There are more than 300 subtypes of SLC transporters have been discovered. The ones that are involved in DDI are organic anion transporting polypeptide, OATPs, organic anion transporters, OATs, organic cation transporters, OCTs, multi-drug and toxin extrusion transporters, MATEs, peptide transporters, PEPs. And those SLC transporters are widely distributed in various organs, such as intestine, liver, kidney, and brain. Transporters that are involved in DDI are summarized in this slide. The major organs that are involved are small intestine, where the drug absorption happens, liver and kidney, where drug clearance happens. The expression of those transporters in intestine intercells, liver hepatocytes, and kidney renal epithelial cells are illustrated here. The ABC efflux transporters, PGP and BCRP, are expressed in intestine, liver, and kidney, as shown in the blue boxes in those drawings. They export drugs out of the cells. If an oral drug is a substrate of PGP or BCRP, the absorption of the drug as intestine is reduced because those transporters pump out the drug from the intercells. The SLC transporters involved in DDI are expressed in liver and kidney, as shown in the red boxes in this drawing. They play key roles in disposition of drugs by transporting the drugs into those cells for metabolism or secretion, therefore affecting the drug's therapeutic effects and toxicity. There are two aspects on evaluating in vitro DDI profile of an investigational drug, determining if it is a victim or a perpetrator in DDI. An investigational drug is a potential perpetrator in DDI if it is an inhibitor or inducer of drug metabolism enzymes or drug transporters. A drug is a potential victim in DDI if it is a substrate of drug metabolism enzyme or drug potentials. Therefore, evaluating the potential of an investigational drug as a substrate, inducer, or inhibitor of the drug metabolism enzymes and transporters in vitro is recommended by both FDA and EMA. Here are some examples of transporter-mediated DDI of the drug that are on the market. This column here are the perpetrator drugs in DDI. This column here are the victim drugs in DDI. Quinidine and dronidone are PGP inhibitors, and rifampicin is an inducer of PGP. As a substrate of PGP, digoxin in this case is a victim drug if it is co-administered with the perpetrator drugs. For example, the digoxin exposure can be increased about twofold by either quinidine or dronidone. The table also shows that some other transporters besides the PGP that are involved in the DDI, such as the OAT, the OCTs, and OATPs. And the US FDA, European EMA, and the Japan PMDA issued a guidance or draft guidance on DDI. US FDA issued the final version of the DDI guidance in January 2020. The EMA issued its draft guidance in June 2012. PMDA issued its guideline in 2018. And those regulatory agencies recommend specific in vitro assays to evaluate DDI. They provide guidelines for drug discovery and the biotech industry on DDI assessment. The FDA recommendations, the FDA recommends DDI assessment in three aspects in its DDI guidance. The three parts are evaluating metabolism-mediated drug interaction, evaluating transporter-mediated drug interaction, evaluating the DDI potential of metabolites. I'm going to focus on the transporter-mediated DDI in today's webinar. In terms of transporter-mediated DDI, the FDA recommends evaluating if an investigational drug is a substrate and or inhibitor of drug transporters. Here is how to decide which assays are necessary. For substrate assessment, if an investigational drug is not highly permeable and highly soluble, it needs to be evaluated in PGP and BCRP substrate assays. If an investigational drug is highly permeable and highly soluble, the bioavailability of the drug is not limited by PGP and BCRP. Therefore, their involvement in DDI is minimum, and the evaluation is not necessary. If an investigational drug's hepatic or biliary secretion is greater than 25% of the total clearance, determining if the drug is a substrate of the major liver transporter OATP1B1 and OATP1B3 is recommended. If an investigational drug's renal active secretion is greater than 25% of the total clearance, determining if the drug is a substrate of the major renal transporters, the O's, the OX, and the MATES, is recommended. Investigational drugs are all investigational drugs are recommended to be evaluated in the inhibition assay in PGP, BCRP, OATP1B1, OATP1B3, the OAT1, OAT3, OCT2, MATE1, and MATE2K. What are the available in vitro test system for for transporter-mediated DDI assessment, and how other in vitro test concentration are determined? For efflux transporters, the inside-out membrane vesicles or polarized cell-based bidirectional permeability assays are used. Because those transporters export their substrate out of cells, it is very challenging to measure the change of the substrate inside the cells. The membrane vesicles are prepared from HEC-239 or insect cells overexpressing efflux transporters. During the membrane vesicle preparation, the vesicles form an inside-out orientation. Therefore, the efflux transporters become uptake transporters in membrane vesicles. The inhibition and the substrate assay can be measured by quantifying the probe substrate or the test compound inside the vesicles, respectively. The second test system for the efflux transporter substrate assessment assay is to use polarized cell-based bidirectional permeability assays, such as KCO2 cells or MDCK cells overexpressing human PGP or BCRP. And KCO2 cells are human colorectal adenocarcinoma cells. They express PGP, BCRP, and other transporters. MDCK cells are Martin-Derby and kidney cells. When those epithelial cells are cultured, they can grow into a polarized format with the apical side facing the median and the basolateral side facing the cell culture vessel surface and to the other cells. They form very tight junctions between each cells. Those characteristics make those cells an ideal test system for permeability and transporter assays. For the uptake transporters, hypothesize our transporter overexpressing cell lines such as CHO, HEC293, MDCK, or other host cells can be used. How is the test concentration determined in the in vitro transporter-mediated DDI assay? Based on the FDA recommendations for the substrate assessment assays, the in vitro test concentration should be in the range of the clinical relevant concentrations of the investigational drug. For the inhibition assay, the test concentration should be greater than 10% of the dose divided by 250 milliliter in apical intestinal transporters, such as PGP and BCRP. The test concentrations should be 10 times of the unbound Cmax in renal transporters, such as the OATs and OX and MATEs. The test concentration should be 10 times of the unbound maxima hepatic inlet concentration in hepatic transporters, such as OATPs. In addition, the test concentration may be limited by aqueous solubility, non-specific binding to the cell culture vessels, and cytotoxicity of the investigational drug in those in vitro test systems. To evaluate if an investigational drug is a PGP or BCRP efflux transporter substrate, two test systems can be used. Polarized cell-based bidirectional permeability assay using KCO2 cells or MDCK cells overexpressing human PGP or BCRP and cultured on semi-poral filter in transvial plates. Our inside-out membrane vesicles prepared from HEC293 or insect cells overexpressing PGP or BCRP. In the polarized cell-based bidirectional assays, KCO2 or MDCK cells are cultured on semi-poral filter in transvial plate, as shown in those drawn here. And after the tight junction monolayer is achieved, the test compound is added on either A side or B side with or without PGP or BCRP specific inhibitors. It is recommended to use more than one PGP inhibitors when using KCO2 cells in substrate assessment assays to determine the specificity of the efflux, because KCO2 cells express multiple efflux transporters. And the amount of test compound in both donor and receiver side is quantified by mass spectrometry or by radiometric assays if radio-labeled compound is used. The apparent permeability of B2A and A2B and efflux ratio, which is the ratio between apparent permeability of B2A versus A2B, are calculated. If the efflux ratio is equal to or greater than 2, and the inhibitor decreases the efflux ratio more than 50%, the compound is a PJT or BCRP substrate. The membrane vesicle assays are relatively straightforward. The membrane vesicles prepared from transporter-expressing cell lines with and without PJT or BCRP specific inhibitors or from empty vector transfected cells are incubated with the test compound. The amount of the test compound inside the vesicle after the incubation is quantified. The compound is considered as a substrate if the uptake of the compound in the transporter-expressing vesicle is 2 times higher than the empty vector transfected vesicles, and the inhibitor inhibits more than 50% of the uptake. The in vitro uptake transporter substrate assessment assays are very similar as the membrane vesicle assays for efflux transporters. As illustrated here in the drawing here, 3 assays are conducted in each substrate assessment assays. Incubate transporter expressing cells with the test compound. Incubate transporter expressing cells with tested compound and transporter specific inhibitor. Incubate empty vector transfected cells with the test compound. After incubation, the amount of compound inside the cells are quantified by mass spectrometry or radiometric assay. The test compound is a substrate if 2 criteria are met. First, the uptake of the compound in the transporter expressing cells is greater than 2-fold of the compound uptake in empty transporter transfected cells. And second, a non-inhibitor can decrease the compound's uptake to less than 50% at a concentration at least 10 times that of the Ki of IC50 of the inhibitor. If a test compound shown as the green dot here is a substrate of the transporter, it will accumulate inside the transporter expressing cells through the activity of the transporter, but not in the empty vector transfected cells because they are now transporter expressed in these cells. When an inhibitor is added, the amount of the compound accumulated inside the cells are reduced because the activity of the transporter is inhibited. And the first method to evaluate inhibition effect of a test compound on efflux transporter is using a membrane vesicle. In this assay, membrane vesicles are incubated with transporter-specific probe substrate and test compound at single or multiple concentrations, as shown in the drawing here. The green dots are probe substrate, which is moved into the vesicle by the transporter. The red dots are the test compound. If the test compound is an inhibitor, the amount of probe substrate inside cells is decreased. ATP is added to activate the ATP-dependent efflux transporter activity. AMP is added as background control. After incubation, the amount of probe substrate inside the vesicle is quantified and used to indicate the inhibition effect of the test compound. The second method to evaluate inhibition effect of the test compound on efflux transporters is using polarized cell. Polarized cell-based bidirectional permeability assay. We can use either MDCK cells or expressing human PJP or BCRP or KICO2 cells. MDCK cells are originated from dog kidney. They express endogenous canine PJP. In order to eliminate the interference from the canine PJP, recently the canine PJP knockout MDCK cells are engineered. and human BCRP, our PCRP, are stably expressed in those canine PGP knockout cells. Those cells provides a more accurate determination of PGP and BCRP inhibition. The KCO2 cells express endogenous PGP, BCRP, and other efflux and uptake transporters. PGP and BCRP function can be isolated by using transporter-selective probe substrate in the assays using KCO2 cells. As illustrated in the drawing here for a B2A permeability assay, the PGP or BCRP are expressed on the apical side of the cells. The probe substrates, which are the green dots in the drawing, is added in the basolateral side. The probe substrate is moved to the apical side by the transporter. If a test compound, which are the red dots here, is an inhibitor, it will decrease the amount of probe substrates in the apical side in this assay. And Zhao, HEC, or MDCK cells overexpressing specific transporters are used in inhibition assessment of uptake transporters. The cells are incubated with transporter-specific probe substrate and test compound at single or multiple concentrations. After the incubation, the amount of the probe substrate is quantified by either mass spectrometer or radiometric plate reader. Pre-incubation of the cells with test compound is recommended because research showed that for some inhibitors, the inhibition potency is time-dependent. For instance, report shows the inhibition of OATP1B1 by cyclosporine is time-dependent. Using clinical relevant probe substrate is recommended because it has been shown that some inhibitors are substrate dependent. As shown in the drawing here, the probe substrate, which are the green dots here, is moved into the cell by the transporter. If a test compound is an inhibitor of the transporter, it will decrease the amount of the probe substrate inside the cell. Based on the FDA DDI guidance, Eurofins Discovery developed and validated a panel of assays that assess a test compound potential to be a substrate or inhibitor of drug transporters that are involved in DDI. In the substrate assessment assays, the bioanalytical method of the test compound is developed and optimized in the assay matrix. First, to make sure we can reliably detect the test compound in the assay matrix. The substrate assessment assays are validated by using known substrates for each transporter. In the inhibition assays, the probe substrate used in each transporter is based on the FDA recommendations. The bioanalytical method is optimized for each probe substrate in the assay matrix. The Km of each probe substrate is determined, and the concentrations of the probe substrate used in the inhibition assay is less than the Km value. The inhibition assays are validated by the possible controls, which are the non-drug transporter inhibitors for each transporter. We offer two methods for inhibition assays, using fluorescent substrate and measured by fluorescent plate reader, or using probe drug substrate and measured by HPLC-MS. Both assay methods have advantages and disadvantages. The assays using fluorescent substrate are run in 96-well plates, and the plates are read by fluorescent plate reader. It is cost efficient and suitable for high throughput screening with fast turnaround time. However, since non-drug substrates are used in those assays, it may cause false negative results if a compound interacts at a different site on the transporter than the fluorescent substrate used in the assay. On the other hand, the inhibition assay using either drug or endogenous substrate are run in 24-well plates. The drug substrates are quantified by HPLC-MS. It directly addresses the DDI because it uses a clinical relevant drug as the probe substrate. However, it has lower throughput and higher cost. This slide shows the assay conditions and the reference compound assay 50 values from the inhibition assays using fluorescent substrate. As is shown here, Zhao, HEC, MDCK cells overexpressing human transporters are used in those assays. The fluorescent substrate and their concentrations are described here. The reference compounds are tested concurrently with the test compounds. The historical average results from the reference compound are listed here. This is the brief protocol that is used in the transporter inhibition assays using a fluorescent substrate. The assays are run in 96-well plate. Transporter expressing cells are pre-incubated with the test compound for 50 minutes at 37 degrees. The transporter specific fluorescent substrate is added and incubated for 20 minutes at 37 degrees with the test compound. Cells are washed to remove the extracellular fluorescent substrate. The plates are read by fluorescent plate reader, percentage control and percent inhibition is calculated, and the assay 50 value is determined if multiple concentrations are tested. This slide is the summary of the assay conditions and the reference compound assay 50 values from the inhibition assay using prop drug substrate. As is shown here, membrane vesicles, Zhao or HEC cells overexpressing human transporters are used in those assays. The drug substrates and their concentrations are described here. The reference inhibitors are tested concurrently with the test compound. The historical average results from the reference compound are listed here. And this is the brief protocol of transporter inhibition assays using prop drug substrates in stable transporter expressing cell lines. The assays are run in 24-well plates. The transporter expressing cells are pre-incubated with the test compound for 30 minutes at 37 degrees. The transporter specific prop drug substrate is added and incubated for 20 minutes at 37 degrees. Cells are washed to remove extracellular drug substrate and analyzed to quantify the intracellular prop substrate by mass spec. Percent inhibition is reported and assay 50 is calculated if multiple concentrations are tested. We use inside-out membrane vesicles in efflux transporter inhibition assays. Here is a brief protocol of inhibition assays using prop drug substrate in membrane vesicles that overexpress efflux transporters. The assays are run in 96-well plates. Membrane vesicles are pre-incubated with the test compound and the prop substrate for 15 minutes at 37 degrees. ATP or AMP is added and incubated for 1 more minute at 37 degrees to activate ATP-dependent PGP or BCRP activities. AMP is the background control. The membrane vesicles are washed to remove extracellular drug substrate and lyse to quantify the prop substrate inside the vesicles by mass spec. Any uptake measured with the AMP incubation is considered as background and subtract from the ATP condition. Percent inhibition is reported. Assay 50 is calculated if multiple concentrations are tested. We determined the Km values of prop drug substrates on each transporters in our lab. Based on the Km values, we determined the concentration of the prop substrate used in the inhibition assays. The rule is that the prop substrate concentration should be at or less than the Km values in the inhibition assay in order for the assay to be able to detect competitive inhibitors. I will show you some representative dose response curves that we generated in our lab. This slide is the dose response curve in OATP1B1 and OATP1B3 inhibition assays when use estradiol 7-beta glucuronate as prop substrate for OATP1B1 and OATP1B3. And cyclosporine, rifampicin, and ritonavir are tested in those assays. This slide is the dose response curve in OAT1 and OAT3 inhibition assays when use p-amino anhydride as the substrate for OAT1 and ranitidine for OAT3. We tested probenecid, timisatin, and ibuprofen in those assays as inhibitor. This slide is the dose response curve in MATE1, MATE2K, and OCT2 inhibition assays when use metformin as prop substrate. Verapamil, imipenem, pyrimethamine, and aquinine are tested in those assays. We use KCO2 cells bidirectional permeability assay for PJP and BCRP substrate assessment, and use CHO or HEC cells overexpressing transporters for uptake transporter substrate assessment. And this is the summary of the substrate assessment assays. It describes the cell line, incubation time, and the transporter-specific inhibitors used in each assay. I will talk in details of those assays in the next couple of slides. In the substrate assessment assay for PJP and the BCRP, two sets of experiments are conducted. The permeability of the test compound in KCO2 cells with and without transporter selective inhibitors. KCO2 cells are seeded in 96-well transwell plate and cultured to form complete monolayer. Test compound is added to either apical site A site or basolateral site B site with or without verapamil in PJP assays or KO143 in BCRP assays, and incubated at 37 degrees for 60 minutes for A2B assay or 40 minutes for B2A assay. The amount of test compound is quantified from both donor and receiver sites by MS. Monolayer integrity is confirmed by fluorescein permeability after test compound permeability assay is complete. The apparent permeability coefficient of test compound and its recovery are calculated from each condition. The efflux ratio, which is the permeability from the B2A divided by the permeability from A to B, with and without inhibitor are calculated. The compound is considered as a substrate if the efflux ratio is greater than 2, and the efflux ratio can be decreased by an inhibitor for more than 50%. The substrate assay for uptake transporters. In the substrate assay for uptake transporters, 3 sets of experiments are conducted. The uptake of the test compound in cells expressing the transporter with and without inhibitor in host cells that are not transfected. Bioanalytical method of the test compound in the assay matrix is developed and optimized first. Wild type and the transporter expressing cells are seeded in 24 well plates. The transporter expressing cells are pre-incubated with transporter specific inhibitors for 20 minutes at 37 degrees. The transporter expressing cells with and without inhibitor and the wild type cells are incubated with the test compound for 2 and 30 minutes and 37 degrees. The test compound inside the cells is quantified by HPLC-MS. So the uptake ratio, which is the uptake in the transporter expressing cell divided by the uptake in the wild type cell, and the percent inhibition of the test compound uptake by the inhibitor is calculated. And the test compound is considered as a substrate if the uptake ratio is greater than 2, and the uptake can be decreased by an inhibitor more than 50%. We tested some non-drug substrate in our assays, and the results are shown here. We observed robust uptake ratio and more than 50% inhibition by non-inhibitors for those substrates. The results showed that we can correctly identify those non-substrates in those assays. This is the summary of the transporter-mediated drug interaction assay panels offered at Eurofins. we offer transporter substrate assessment assays and transporter inhibition assessment assays. For the substrate assessment assays, based on the membrane permeability and the solubility of the test compound, you can decide if the efflux transporter substrate panel is necessary. Based on the route of clearance, you can decide if the liver drug transporter substrate assessment panel or renal drug transporter substrate assessment panel is suitable for your compound. In the transporter inhibition panel, you can choose the complete panel to meet regulatory recommendations, or you can choose any individual assay to address certain concerns. The inhibition assay can be run at a single concentration or multiple concentrations for assay 50 determination. By offering panels of assays that assess transporter-mediated drug interactions, we hope that we can provide our clients with a useful in vitro tool to evaluate the DDI profile of their investigational drugs and meet regulatory agencies' recommendations. Before we get to the question and answer session, I will give you a brief introduction of Eurofins Scientific, Eurofins Discovery, and ADME-Toxicology services at Eurofins Discovery in the next few slides. Eurofins Scientific is a leading international group of laboratories providing a unique range of analytical testing services to the pharmaceutical, food, environmental, and consumer products industries and to governments. It was established in 1987. It started in food testing, then expanded into environmental testing, clinical diagnostics, and subsequently biopharmaceuticals testing, which is our division. Biopharm is the newest platform on a very successful existing organization. So Eurofins as a whole had over 6 billion euro in revenue last year. There are over 60,000 employees and more than 900 laboratories worldwide. Eurofins Discovery is the Eurofins Drug Discovery CRO services branch. By combining the market-leading drug discovery expertise and capability, Eurofins Discovery strives to provide solutions for every problem you have in your drug discovery program, from medicinal chemistry, in vitro pharmacology, in vitro safety pharmacology, ADME-Toxicology, translational phenotypical services, and in vitro services to customer assays and products. And there are more than 1,500 scientists working at Eurofins Discovery, with 7 locations worldwide located in San Diego and San Francisco in California, St. Louis in Missouri, France, Spain, and the UK in Europe, and Taipei in Asia. We have over 2,000 active clients who are using our services for their drug discovery projects, have more than 85 patents. We offer more than 7,500 unique assays and generated over 30 billion data points, and have more than 67 IND filings for our clients. We handle over 14,000 projects per year. We do not have any competing internal drug discovery program. Our commitment is to our clients. We have been providing ADME-Toxicology services in drug discovery for over 25 years. The ADME-Toxicology services are provided at our St. Louis, Missouri location, as well as in Spain in Europe. Eurofins Discovery has extensive experience in consulting, designing, and conducting in vitro ADME and toxicology testing programs for thousands of clients in the pharmaceutical, biotech, consumer health, as well as for leading academia and government institutions. We offer over 400 in vitro ADME assays and over 100 in vitro safety pharmacology toxicology assays, which provide you with a comprehensive solution for assessing drug bioavailability, metabolism, drug-drug interaction, toxicity, and safety. Why choose Eurofins as your partner? Our scientists have over 40 years of industry expertise in drug discovery. We provide quality data with fast turnaround times. We have more than 2,500 active clients worldwide using our services for their drug discovery projects. Our scientific team have exceptional knowledge and experience in drug discovery. Our services are flexible in order to provide customers solutions to fit your testing needs. We provide comprehensive services to help our clients to progress their projects from ideas to preclinical candidates. Last but not least, I'd like to acknowledge my colleagues who are part of the DDI innovation projects at Eurofins Discovery. We could not develop and validate those assays without the dedication and the contributions from those team members. This concludes my presentation. I'm happy to answer any questions you have. Thanks for your time.
Thank you, Yong, for your excellent presentation. We now have the pleasure to introduce our question and answer session, where today's speaker will answer all of your questions. Don't forget, you can still submit your questions using the question panel in the menu on the right. So let's take a look at our first question. What is your opinion on the model systems used for preclinical DDI assessment? What does the guidance document recommend in terms of cell lines or other in vitro systems? What would your recommendation on a system be?
Right, thank you for the question. So the test systems are based on the available published researchers. Some systems are high throughput friendly, ideal in the industry settings, and some are more suitable for academic researches. The FDA does not recommend certain test systems over others. However, it emphasizes that every test system needs to be fully validated and compared with the published results from other test systems. In my opinion, the membrane vesicles for efflux transporters and the human transporters stably expressed in Zhao, HEC293 or MDCK cells for optic transporters are industry-friendly methods due to the nature of the assays, such as the throughput, low cost, and the reproducibility of the data. Of course, each test system has its own advantages and disadvantages. When considering which test system to use, the sponsors need to take into consideration of the profile of their test compound, such as the permeability, solubility, and cytotoxicity of those compounds.
Thank you. Okay, so moving on to the next question. Drug metabolites can often act as a substrate or inhibitor for other transporters. In your slide, you showed, for example, N-methylquinidine as substrate for P2B or estradiol beta-glucuronide. In your opinion, when would it be important to screen for DDI of metabolites of drugs?
It is a great question. So as I mentioned in my presentation, FDA recommends assessing DDI in three aspects. First, evaluating drug metabolism enzyme-mediated DDI. Second, evaluating transporter-mediated DDI. And the third, evaluating metabolites in the mediated DDI. So FDA recommends that metabolites with significant plasma exposure or pharmacological activities may need to be evaluated for its DDI potential. And if in vitro DDI assessment is warranted for metabolites, FDA recommends using a synthesized or purified metabolite standard in those assays. So the synthesized or purified metabolite standards will be tested as a test articles in the assays I discussed in this webinar if necessary. And if metabolites contributes more than 50% of the overall activity pharmacologically, it is recommended evaluating its contribution to DDI as a substrate of the metabolism enzymes and transporters, because if it is a substrate, it could be a victim in the DDI, and the pharmacology activity of the drug could be impacted. If the metabolite is suspected as an inhibitor of the drug transporters, it needs to be evaluated as an inhibitor in vitro. However, if in vivo DDI study are planned, the in vitro assessment of metabolites as enzyme or transporter inhibitors may not be necessary because the inhibition potential of the metabolites would be evaluated in vivo along with the parent drug.
Thank you. Okay, so moving on to the next question. Firstly, they say thank you for the webinar. Their question, this seems to be more focused on small molecules, but I was curious, for therapeutic proteins, how much of a concern is DDI and when would an assessment be necessary?
And there are two situations of therapeutical proteins in DDI. So the potential for a DDI between a therapeutical protein and the small molecules, or between therapeutical proteins. And FDA recommends considering the mechanisms of a potential DDI, taking into account the pharmacology and the clearance of the therapeutical protein, as well as any co-administered medications in the patient population. So FDA published a draft guidance of DDI for therapeutical proteins around the same time as the DDI guidance for small molecules, as we discussed in this webinar. In the draft guidance, DDI discussed some situations in which the assessment of the DDI potential of a therapeutical protein is necessary. For example, if the therapeutical protein is a pro-inflammatory cytokine modulator, or the therapeutical protein is a pro-inflammatory cytokine, DDI potential of the therapeutical protein should be evaluated in metabolism enzyme-mediated DDI, because the pro-inflammatory cytokine can downregulate the expression of the CYP enzymes. So I did not discuss about the enzyme-mediated DDI in this webinar today. So if the therapeutical protein is not a pro-inflammatory cytokine or cytokine modulator, and if there is a known or suspect mechanism for DDI, the DDI assessment is recommended in this case.
Okay, great, thank you. Next question. For your validated assays, what is the criteria used for signal to noise on your LC-MS assays? Does this relate to uptake ratio?
The assays that use LC-MS to quantify the test compound in the cells are the assays that assess if a test compound is a substrate of the transporters. So we first developed and optimized LC-MS method to quantify the test compound. We have stringent criteria to meet in method development and optimization in order to have a reliable MS quantification method. In the method development and optimization phase of the study, we test the three concentrations of the test compound in the assay matrix, normally at 30 nanomolar, 100 nanomolar, and 1 micromolar. We require that the peak area response should be proportional over the concentration range with acceptable signal-to-noise ratio, which is greater than threefold above a background at the lowest concentration, based on a blank injection. So however, this signal-to-noise ratio is not the same as the uptake ratio in the transporter assay. The signal-to-noise ratio is to demonstrate the robustness and how reliable of the MS quantification method. The uptake ratio in transporter substrate assessment assay is the ratio between the uptake of the test compound in transporter expression cells versus the uptake in empty vector transfected cells. It is used to determine if the test compound is a substrate of the transporter. So we need an acceptable signal-to-noise ratio of MS quantification method in order to measure the uptake ratio in the substrate assays.
Thank you. In your experience, when are researchers conducting this type of DDI assessment?
So I think most of the researchers conduct in vitro DDI assessment in preclinical phases in drug discovery, and some assays can be used to discover the mechanism of DDI after the in vivo testing and the clinical trials. But some of the assessments, such as the CYP inhibition, CYP phenotyping, and PGP, BCRP substrate assessment can be done earlier in the drug discovery phases because the results from those assays can provide information on bioavailability of the test compound.
Thank you. In your experience, are inductions of transporters similar to CYP induction a regulatory concern for DDI?
There are some non-DDI due to induction of transporters, such as the impact on digoxin exposure by rifampicin, and is due to the induction of the PGP by rifampicin, as I mentioned in the webinar earlier today. So, however, there is not enough conclusive research on the contribution of transporter induction to DDI. The regulatory agency do not issue a recommendation on conducting a standard transporter induction assessment of an investigational drug for DDI evaluation at this time. So I don't think there's a need, unless you have very solid evidence that the induction of the transporter in the DDI, I don't think there's a need to evaluate the induction potential of investigational drug for DDI assessment.
Great, thank you. Most transporters have many different drug substrates. How do you choose a probe substrate in transporter inhibition assay?
And this is a great question. So the inhibition can be substrate dependent. So this is mainly due to different binding sites for different substrate on the transporters. And the regulatory agencies recommend that determining the inhibition potential of the investigational drug with a probe substrate that may also be used in later clinical studies. Alternatively, you may use a probe substrate that usually generates a lower IC50 for non-inhibitors in order to avoid underestimating the interaction potential of the investigational drug.
Thank you. How do you position the host 3342 diefflux assay for BRCP compared to the inside-out vesicle or polarized cell A to B, B to A assay?
So as I mentioned in my presentation today, so the Herx 3342 dye assay is the fluorescent substrate assay that we use in the transporter inhibition assays. So usually the fluorescent substrate inhibition assay is positioned at the early stage of the drug discovery for the screening purpose. They give you an indication that if a test compound is an inhibitor of the BCRP transporter in this case. However, if the stage of your drug discovery is close to the clinical at the end of a preclinical phase, I would recommend that we use the inside-out vesicles or the A to B, B to B, A to B, B to A permeability assay for the inhibition assessment, because in those assays we use the drug probe substrate, so that gives you a direct measurement of the drug-drug interaction.
Thank you. The CACO2 cell's monolayer periplasmic space. The intercell contacts are not similar to that of intestine cells, for example. How representative is this model to absorption?
I think the CACO2 cells are the CACO2 cell, like I mentioned in my presentation, it is the cancer-derived cells from human. It is a very good system for evaluating the permeability from intestine, and also we can use this system for the drug transporter assays, as I mentioned here, because it expresses human PGP, human the BCRP, and some other efflux and the uptake transporters. It is considered as a good system, but however, as I mentioned, there are multiple drug transporters expressed in CACO2 cells. So sometimes there's a challenge to isolate the particular transporter that you want to study. For example, there's some research showing that there it's a kind of crosstalk of the probe substrates between PGP and the BCRP. So in that case, the MDCK cells overexpressing either PGP or BCRP may be a better system to isolate those drugs, the drug transporters in an independent system.
Thank you. We will address any questions, follow up by email of anything that's been asked, and not been asked right now in the live Q&A session. But unfortunately, that's all we have time for today. So please join me in thanking Yong for his time today. As you leave the webinar, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.